So just to recap, we've talked about DNA and what it is. We also talked about transcription, how the information on one gene of DNA can get copied onto a messenger RNA. Now we're going to talk about how that message gets translated into making a protein. So I just want to start with an analogy. DNA, you can think of like a cookbook. I don't know if any of you cook, but it has the instructions. Messenger RNA is basically equivalent to making a photocopy of that. So say you had a book you couldn't take with you, you made a copy of that page, the one page in the whole book. That's what mRNA is, and we made that with transcription. Now will be the process of making the thing that the book page is instructing us on. For us, those are going to be proteins, amino acids. More so, we'll put a string of amino acids together to end up with a whole protein, like the cake in this example. So translation, this is the assembly of a protein using genetic instructions. It makes sense, so transcription was making a copy. Translate, it's like translating a language. We're going from DNA language to protein language. So, decodes information from mRNA to assemble an amino acid chain, and an amino acid chain is a protein. So, nucleotides of the messenger RNA, which is shown up here, an example of that. These are read as triplets, which we call codons. So a triplet means 3 bases at a time. So these 3 bases would be one codon, these would be another codon, and so on. So you can see that nicely here in this example. So AUG is one codon, and you'll get a specific amino acid from that. Same with AAC. That's one codon. We'll get a specific amino acid from that. So here's another example of how to see that. So a codon you can think of as a 3-base or a 3-letter word that codes for one amino acid. So 3 nucleotides of the messenger RNA equals one codon equals one amino acid. So all the codons put together, all the amino acids put together gets you to the protein. And it can take hundreds to thousands of codons, which means hundreds of thousands of amino acids to make up one protein. So translation, here's sort of a diagram of how this is working. You can see your messenger RNA here. So this occurs at a ribosome, which is the blue structure shown here. This is outside of the nucleus in the cytoplasm, or if you remember, you could see it on the rough endoplasmic reticulum. And we're going to require a few things. So you need your messenger RNA that has the instructions. Your ribosome is the location. And the tRNA, which are these little orange guys that sort of look like a T, this translates DNA language into protein language, meaning it reads nucleotides in order to put together amino acids. So they're 2 different types of macromolecules with different types of monomers. Alright, so the steps of translation. So first, messenger RNA is moved through the ribosome, reading one codon at a time. The tRNA reads the mRNA codons and adds the amino acid that it signifies, adds that to the protein chain. And then finally, once you get to the end of the protein, a ribosome is going to read what we call a stop codon, and that signals that the protein's finished and it'll be released and go off to do its protein things. And then that ribosome is free to make more proteins in the future. So mRNA gets there and travels through, tRNA reads the mRNA one codon at a time to add one amino acid at a time, and then we're left with a full protein. So here's sort of what that looks like. You can see the mRNA sneaking through, this one tRNA reading that one codon. So you can see it reads it with sort of a matching sequence, and it adds one single amino acid. And then a second one will come in, and that amino acid will get added to the other one to start off that chain. And then you can see after that's done, another tRNA will come in to read the next codon to add the next amino acid. And that just keeps going and going until you end up with this full amino acid chain. And again, once it reaches a stop codon, it detaches and it's complete. So tomorrow we'll talk about how exactly we know which amino acid goes with which codon.